We are now seeing a massive increase with more than 200 clinical trials currently ongoing. Especially the COVID-19 pandemic was the biggest driver of this technological innovation. If there is an issue in this cascade resulting in, for example, too much or an incorrect protein, oligonucleotides can inhibit specifically these pathways. You need significant expertise to turn ugly raw spectrum into precise results. So you need to have the expertise on your instrument and also on your matrix to know how you can develop a good method for your issue.
Hi, everyone. Welcome to Expert Insights. I'm Cindy Dubin, Managing Editor of Drug Development and Delivery, and I am here today with Dr. Melanie Zerulla-Wernitz, Head of Analytical Science Laboratory at Vetter. She is responsible for a diverse team of excellent scientists who combine various natural and applied sciences. Thank you for joining us today, Melanie.
Thanks for having me.
I know you have a lot of great insight to share with our viewers today about LC-MS technology and methods, so I thought we could just jump right in.
We can do.
Perfect. So we have seen a strong shift towards oligonucleotide therapies. Could you briefly outline the current state of that market?
While the first approval of therapeutic oligonucleotide occurred in 1998, we are now seeing a massive increase with more than 200 clinical trials currently ongoing. So infectious diseases in general, but especially the COVID-19 pandemic was the biggest driver of this technological innovation. And these molecules, the oligonucleotides, are applied across a wide range of medical fields. So from metabolic disorders and oncology up to immunology.
So how do these molecules function within the body to treat a disease?
Oligonucleotides interfere with one of the, let's say, most famous cascades of biology, where the DNA from the cell nucleus is transcribed to mRNA, which is then translated into proteins. So if there is an issue in this cascade resulting in, for example, too much or an incorrect protein, oligonucleotides can inhibit specifically these pathways. So for example, if you look at an antisense oligonucleotides, this binds to an existing mRNA to trigger its degradation, while, for example, aptamers form 3D structures and bind highly sensitive to target proteins and prevent their bioactivity.
So then what does the structure of the molecules mean for their analysis, and what makes them so difficult to work with as a drug in a lab?
So structurally, the oligonucleotides consist of nucleotides, and these consist on a phosphate, a sugar, and a base. And this is something like pearls on a string. And in the therapeutic use, they are typically very short, around, let's say, 20 nucleotides in length, and very often double-stranded, forming a helix structure via the Watson-Crick base pairing. But in the natural environment, so in the patient's body, this makes them very unstable, because our cells in our bodies have evolved a view for foreign DNA or RNA as a viral threat. So there are specific enzymes everywhere in our body that degrade them. And furthermore, the oligonucleotides have very poor cell penetration ability, and they are not naturally tissue specific. So let's say they are somehow blind in the body. And to solve this, there are multiple chemical modifications out there to ensure the drug reaches the target organs, for example, like the liver. But these modifications can make the analytical testing even more challenging.
So then why is LC-MS particularly suitable for oligonucleotide analysis?
Because the analytics combine a very effective separation ability with a very high specific molecular mass detection. And oligonucleotides differ by very small mass changes. And with LCMS, the sequence can be confirmed, the sequence of the oligonucleotides, but also the modifications, these chemical modifications I mentioned before, could be localized, or you can see somehow fragmentation patterns, for example.
So then how do you decide which specific system to use when dealing with these complex molecules?
So there's a wide range of different LCMS out there. And the answer is very difficult because it really depends entirely on the analytical question you want to answer. So, for example, for very robust identity testing or simple quantification, a single quad would be your choice. However, if your daily business in your lab is a intensive quantification, then the triple quad is the state-of-the-art instrument. But in our development lab, we choose a QTOF because it allows us to handle changing questions every day, which is the normal situation in a development lab. For example, as structure determination and testing for investigational purpose.
So are there specific technical challenges when analyzing oligonucleotides on an LCMS?
Yes. So one of the biggest issues is that the phosphate groups, which I mentioned, which are a part of the nucleotides, they are negatively charged. And this leads to a lower ionization efficiency, and it could end up in multiple charge stages. And additionally, you could have an issue with adduct formation due to alkali metals like sodium or potassium, which is normally in your matrix very often. And this can broaden the peaks and reduce the sensitivity. So you need significant expertise to turn, let's say, ugly raw spectrum into precise results. So you need to have the expertise on your instrument and also on your matrix to know how you can develop a good method for your issue.
And how does LCMS support early stage development of oligonucleotides?
We use our LCMS to detect and quantify both known and unknown impurities, even in trace amounts, because it's so sensitive. It is also essential for monitoring degradation products during manufacturing or stability studies, and to confirm the completeness of the chemical modifications. But also simple tasks like identification, sequencing, and quantification could be conducted reliably with our LCMS.
So speaking of reliably, if a lab doesn't have an LCMS, are there other reliable ways to identify these molecules?
Yes. We also utilize a melting point method using UV-Vis spectrometry. By measuring the hyperchromicity effect as a duplex melt. So if you have a double-stranded DNA or RNA, if this melts up to 2 single strands, then we get a melting curve that provides a very high specific and selective parameter for the identity. And this is an excellent alternative if no LCMS is available, for example, in a QC laboratory.
Well, Melanie, thank you so much for all of this insightful information. Vetter is a highly respected CDMO, and I know our viewers learned a lot from you today. So thank you so much for your time.
Thank you so much for having me for that interview.
And thank you all for joining us today on Expert Insights.